Hello, everyone, and welcome to today's webinar. Learn how to maximize Kingfisher instruments to automate DNA, RNA, proteins, and cells for all applications. I am Marie Stone of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Thermo Fisher Scientific. To learn more, visit thermofisher.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. This webinar is educational and thus offers free continuing education credits. Please click on the Continuing Education window at the bottom of your screen to obtain your credits. I'd like to now welcome our speakers. Dr. Suraj Patel, EMEA Sample Preparation Business Manager, Thermo Fisher Scientific. Emily Zeringer, R&D Staff Scientist, Sample Preparation Biosciences, Thermo Fisher Scientific, USA. And Dr. Ketil Pedersen, Manager and Staff Scientist, Thermo Fisher Scientific. Welcome. You may now begin your presentations.
Thank you, Marie. Okay, so thank you very much for your time today. So the agenda for this meeting is going to be, we're going to first focus on methods for sample purification, then we're going to focus on benefits of automated workflows, going to go into the SARS-CoV-2 isolation and beyond, and then moving into transitioning between these applications, and then there's obviously a Q&A session at the end. So we're gonna talk about methods for sample purification, and as you saw at the beginning, this was focused on the Kingfisher instrumentation. So the story kind of begins with nucleic acid extraction. So this next slide just shows you three common types of nucleic acid extractions that customers will normally do. You've got spin columns, organic extractions such as trizol or phenol, and then you've got automated systems as well, because there are other ones out there on the market. So this just kind of points out the challenges that people tend to face with these systems. So with the spin columns, just to highlight a few, it's very labor intensive and quite time consuming. With organic extraction, consistency can be a problem and working with difficult toxic agents. And then with automated systems, you tend to be locked into the protocol that the instrument gives you, right? Or consumables such as cassettes and things like that. So you don't get much flexibility for your sample type or your protocol. So what we wanted to do at Thermo Fisher is we wanted to create a solution for some of these pain points, which involves magnetic beads and automation. So believe it or not, this solution's been around for 22 years, and that's the Kingfisher product line. And you can see here a timeline that we've put up of when the first Kingfisher was introduced in 1999, and then subsequently other instruments after that. I just want to focus on the Kingfisher Flex in the middle of your screen, the Kingfisher Duo Prime, and the Presto. They're the current three instruments out on the market, which are the most popular. And then just recently this year, we've launched the Kingfisher Apex in 2021, and that's already doing really well and is a big upgrade to the for how the systems are going to be moving forward. So today we've got 17,000 instruments out in the field, and just at the bottom there you can see in terms of our applications, one of the biggest ones we were thrown into is the SARS-CoV-2 testing because of the fact that we can do RNA extractions on our systems. So just to give you a little insight into how the Kingfishers work, it works on two main plate formats. The first is a 96 deep well, and it has a working volume which must include your sample, your buffer, your beads, everything from 30 microliters up to 1 mil. So this is mainly designed for high throughput extractions. We've got another plate format, which is the 24 well plate, and that allows you to have a higher volume. So you're going to work from 0.2 mil up to 5 mil. So this is really good for the higher volume work, but you do compromise on throughput because we can only do 24 samples at a time. To put it into some kind of real perspective, if you were extracting DNA from blood, the first 96 well plate would be for 50 microliters of whole blood to 400 microliters of whole blood. And then if you want to go up to, say, 1 mil to 5 mil, then you'd be using the 24 well plate. So that's kind of how that works. There are different heads available. So we talked about the 24. and the 96. There's also a smaller 96 well head for low volume, and there's also another 96 well head for PCR plates. So we can use them on the system as well. Just to go into a little bit more detail about the individual systems and how they compare to each other, I'm not going to go through the whole slide, but I will highlight that the Duo Prime is focused on that compact benchtop instrument, right? So for low to medium throughput, and when you're working on 96 well plates, they extract 12 samples at a time. When you go to the 24, you're extracting 6 samples at a time, and this is really good for research use only. When you want that next step up in throughput, you've got the Flex. So this is more the medium to the high throughput, and you can see the samples per run. It operates on the whole 96 well plate at a time, or a whole 24 well plate. So you can get much higher throughput in your extractions. And then you've got, and both of these instruments work on the Bind-It software, which is a separate software that comes and allows you to actually change and optimize your protocols if you wish. I'll come back to the Apex. The Presto is another version of the system, but this is made for liquid handling instruments. So if you want a complete automation solution where you're not putting any samples or liquids into the plates yourself, but you want a robot liquid handler to do that, you bring in the Presto, and that will do all of that for you. And again, it works on 96 and 24. So the Apex is kind of an advancement on the Flex, and you get some extra features such as being able to have 2 heads on the machine, UV light, and also a cooling function. The rest of them have heating, but the Apex has a nice cooling function for certain applications. So we did a survey and looked at our customers and tried to narrow down what our customers are using the instruments for. Most of them are for infectious disease testing, and kind of, I guess a lot of that falls into COVID testing. But then we've also got others for virology, oncology, sexual health, and also other applications such as bacterial detection, and genomic DNA and RNA extraction is quite popular as well. So I want to talk about the workflow, and I want to kind of just introduce how we create this high throughput automatable workflow with the Kingfisher and using our Dynabeads. So the next slide just focuses on those key points. So with our combination of the instrument and the beads, we get better increased sensitivity, better significant time savings because it's automated and you don't need to be using it at the bench, and then increased productivity. And then here you can see that using the Dynabeads, when you're using it in a manual situation or with a magnet, you're liquid handling. So if you're familiar with Dynabeads, you're introducing your beads, they bind the target, you're then holding the beads, washing them, and then finally removing your target out in solution while holding the beads. The way the Kingfisher works, it turns that on its head. What if you could pick up the beads? So with the Kingfisher, you load the plates. So each plate becomes a step in the process. You load the plates onto the machine. The machine will mix the plates, pick up the beads, and move them from one step to another. And eventually in your elution plate, it will elute your target off the beads, pick up and remove those beads, leaving your target behind in the elution plate. So that's essentially how the Kingfisher works. And in this case of the Apex, you've got a turntable for each of your plates. Okay, so let's take that to our most popular application, which is SARS-CoV-2. So this just really quickly shows you the Thermo Fisher workflow for SARS-CoV-2. And obviously, we've got products for receiving samples, real-time PCR and analysis, but we're focusing on the middle part, which is preparing samples. And this is a slide that just shows you our workflow with our MVP2 kit. And you can see starting from the left, whether you've got an inactivated sample or a live sample, we've got protocols for both. We can inactivate with our binding buffer and then go straight into our extraction. So that works really well, and that extraction is performed in 22 minutes. So what I want to kind of really focus on this presentation is going beyond SARS-CoV-2. If you've already got the instrument and you're already doing SARS-CoV-2 extractions, what more can you do with the system? That's kind of what I really want to focus on. And this slide just looks at it because I want you to think about it in 2 ways. One from the sample perspective and one from an application perspective. So currently, if you're using it for SARS-CoV-2, you're going to be using a nasal pharyngeal swab. So that is one kind of method that you can use. But we've actually got protocols for the MVP2 kit for different types of samples. So whole blood, saliva, plasma, serum, all of these can be used on the same system that you're already using and be able to extract. And then we can actually go beyond that. We can actually go to different other types of sources. So you can see animal health, plant and soil, and food safety. And then we can also do other targets such as cells, stool samples, even FFP samples. So we've got kits within Thermo Fisher that can do that. But the key thing to understand from a sample perspective, you can go way beyond what you're currently using it for, even to those far fringes of plant and animal health samples. To take that another step further, like I mentioned, is thinking about it from applications point of view. So we're currently doing viral testing at the moment for SARS-CoV-2, but you can also use it. You can see on this deck from the left-hand side is focused on nucleic acids. So you can see we've got beads and kits for PCR cleanup, high molecular weight, and then other types of RNA extractions as well. But we've also got beads for protein isolations. So you can do Luminex, immunoprecipitation, Smart Digest, which is a trypsin digest, and we can also do cell isolations as well. We can also work with custom beads because you can change the protocol to what you need it to be. And all of these applications are ready for downstream, such as digital PCR, NGS, chromatography. So you can see from this slide and the previous one, the system opens up a lot more sample prep than where you started with, and hopefully new things that you can think about doing with your system. This slide just shows you various kits that we have. So we have various kits to go for DNA, RNA, total nucleic acid, proteins, and cells. And then I kind of want to talk about that next step beyond that. So we were looking at SARS-CoV-2 from nasopharyngeal swabs, but we've also done a lot of work with customers on wastewater samples. So these next few slides just talk about the importance of wastewater. So the idea is to use wastewater surveillance testing, and it can be for various targets. Polio is quite one that's quite common, but we were focusing on SARS-CoV-2 and basically allowing customers to use our workflow in testing the wastewater. So we've got two types of protocols that I wanted to highlight, and they're using our wastewater nucleic acid enrichment kit, which is highlighted here. And the first protocol focuses on 1 ml of your wastewater sample directly into our kit. So you just take 1 ml, go straight into our kit, and do the extraction, and you can get some quantification. But then we've got some data here showing you that as well. So you can see that we spiked in some controls and did some quantification with qPCR on that. And then the next step is a protocol that's again using the Kingfisher, but we're using a viral concentration with some magnetic beads and using a much higher input of 10 mil. So you can see from the next slide the data showing that we're getting consistent extraction and good CT values for SARS-CoV-2 in 10 mils of wastewater. So if you were interested in doing that, that's another thing you can do with our Kingfisher. But like I said, we can go beyond that. So I just wanted to highlight one of those applications, which is oncology, and I wanted to focus on detection of cell-free DNA. So this is cell-free DNA is something that oncology researchers are looking at, and we're looking at that because in oncology we want to be able to take a kind of liquid biopsy from a patient, and that is what we're kind of focusing on here. So we're taking a blood sample and we're trying to see if we can look at the cell-free DNA coming from the tumor and be able to diagnose or do some therapeutic treatment with those patients. So the next slide here is, excuse me. So the next slide here is showing the cell-free DNA kit that we use for this. And this kit will be used for isolation of cell-free DNA from serum, plasma, or urine. And the key thing to understand when you look at the traces, if you do a quantification of just concentration, it'll end up quite low to what you'd be expecting. But that's because our purity is much higher when you run it on a bioanalyzer. You can see that our cell-free DNA peak is much more concentrated compared to the genomic DNA, which is decreased. So this is something that's quite normal with our kits. So our purity is much higher. And then this is just showing you some digital PCR data where they were comparing the extraction from FFPE samples and also liquid biopsy to see if there's any differences in the genetic profile. And then finally, for taking it that step further, as well as cell-free DNA, some people are looking at circulating tumor cells. And I know this can be quite difficult to isolate. So there's a publication which we worked with a customer, Rui, in Düsseldorf. And what we were doing is trying to see, can we isolate circulating tumor cells from using the Kingfisher and some targeted beads. And you can see from this data here that I'm just showing, it worked really well. Not only did we isolate, we fixed and stained the cells as well. So it's worth looking at that paper. Okay, so hopefully hand over to Emily for this part.
Yes, thank you, Suraj. And thank you to our listeners again for coming into the webinar. So I'll be taking over and covering another application, switching gears a little bit to genetic testing. Genetic testing, of course, focusing on DNA extraction with the downstream application examples of genotyping and also exome sequencing workflows. And so traditionally, genetic testing has been performed with whole blood and buccal swab samples. However, in recent years, saliva has really emerged as a popular collection method. And for several reasons. One, it's very automation and high throughput friendly, being a liquid sample. It's also a much simpler collection method. Saliva is often self-collected. And there's two ways really you can collect it, either as a raw saliva sample into a tube or collecting into a stabilization reagent, which preserves the nucleic acid in the sample and allows you to store it both for longer and to store at ambient temperatures. So in addition to simple collection, saliva is also, it fits very nicely into an automated workflow. The collection tubes are barcoded and in standardized sizes, and so function in liquid handling as well as cappers and decappers. They also allow quick delivery of results, being a easily accessible and easily processed sample. In addition to these benefits, which are very useful for genetic testing, they also have a benefit which has become of interest to pathogen detection, is that it is low cost, but also especially the stabilized saliva provides some viral inactivation, allowing the samples to be processed outside of highly, BSL-3 or BSL-2 labs. So, it makes saliva a nice sample and kind of unique in that it covers all of this compared to buccal swabs and whole blood. And we actually do have, for both the raw and the stabilized saliva, our Specimex devices provide an option for this. So if you have questions about that, please be sure to ask in the Q&A. So as I said, saliva has become of great interest, kind of an emerging sample type people would like to use. But when using a new sample type, you of course want to make sure that it compares well back to the original ones and make sure that they match in concordance and performance. And so here we actually used our MagMax DNA Multisample Ultra 2.0 kit combined with the Kingfisher Flex in order to conduct a concordance study. And here we compared whole blood, buccal swab, and saliva, 3 sample types from donors. So they were matched samples. And to the right, you can see as far as for each sample type, kind of their, from, decreasing, discomfort. Whole blood, of course, is the most evasive. Buccal swabs, fairly simple, somewhere in the middle. And then, of course, saliva. It really doesn't get much easier than spitting in a tube. So anyway, we collected these 3 matched sample types from 25 donors, ranging in ages, and then performed our extraction workflow and analysis, both for qPCR for genotyping as well as sequencing. And so here you can see we performed the genomic DNA extractions using our Ultra 2.0 kit and our Kingfisher Flex. They quantified and checked quality of the DNA using the nanodrop and the qubit instruments and kits, and then looked at 2 different types of downstream analysis. We looked at sequencing using the AmpliSeq exome kit, and then we also looked at real-time PCR with several of the TACMan genotyping assays. And so we performed this for all donors, and then for all of the sample types for each donor. There we go. So here you can see kind of a snapshot of the results. The top graph is showing the genotyping % call rates for both the heterozygous and both homozygous alleles for the 3 sample types for all different donors. And these are just targeting 3 specific genes, or SNPs, excuse me. And as you can see, the call rate is equivalent, almost identical for all 3 sample types between the different donors. So showing very good concordance for genotyping. The same if you look at the bottom graph, variant calling concordance. You can see it's all in red, and the red indicates the variants are the same in all samples for all sample types. Very good results indicating all 3 sample types provides the proper results needed. And then here, kind of looking at the overall variant calling concordance between the sample types and between all the different donors. You can see between 22 donors, we have a 94% matched variant call rate between the buccal swabs and saliva, and then greater than 93% for the whole blood to saliva, and then whole blood to buccal swabs. And so based on the study indicating a very high concordance between the different sample types, between the matched sample types between the different donors, indicating that saliva, in addition to having good benefits, also will work in the workflow. And so study was beneficial twofold: one, to show that saliva is a very useful sample type, and 2, to show another way you can use the KingFisher Flex instrument. And then of course, just a few details about the Ultra 2.0 kit. Very versatile, like the Flex instrument. It allows both low sample input up to high sample input using both the 96 and 24 well formats, and is compatible with the other KingFisher instruments in addition to the Flex. So as Suraj mentioned, the Flex allows you you can use the Flex for multiple different research applications. Everything from SARS-CoV-2, which Suraj gave a nice summary on, to looking at different bacteria, also looking at kind of more specific research areas: sexual health, women's health, and healthcare-associated infections. And so we'll actually, the next section, talk about one of these areas, looking at vaginal and urinary microbiota with the Flex and MagMex kits as part of the workflow. And so, so specifically looking at detection of vaginal urinary tract microbiota, the first question is kind of why, would you want to look into this area? Why is this important for research? And just kind of interesting facts. Urogenital infections affect greater than 1 billion women each year. And of these infections, some are associated with additional types of infections like cervicitis and pelvic infections. And so if you're looking more specifically at bacterial vaginosis, it affects 10 to 15% of women reproductive age. And of course, this kind of converts into a huge cost because it can cause pregnancy complications, and so it's just kind of it's something you wish to avoid if possible. And so, by doing research and being able to detect pathogens specific to this to understand can be very beneficial to a large number of women. And so traditionally, for detection of these types of pathogens, culture-based detection methods were used. And although they work very well, they have some drawbacks such as lack of sensitivity. They can be subjective and often require very lengthy turnaround time. As an alternative, in, I guess, the past decade or so, they've started to transition to molecular methods, which have really reduced turnaround time and increased sensitivity and specificity for being able to detect specific pathogens. And so, just to give you more of a graphical representation, between the traditional workflow and then the molecular workflow. So here you can see traditional, you have 2 different sample types. You have your purification with a nucleic acid or protein, or just purifying the different cells, separate for each sample, and then, what type of sample are you looking for? And then, what type of test? And so you can see it can grow exponentially, and kind of a reason why sometimes these results can take quite a long time. Alternatively, we have the molecular workflow, which similar workflow, but simplifies it immensely. So, most of these have been converted into high throughput, even automated. And so you have multiple samples, one method, one downstream output. So simple, comprehensive, and allows for targeted analysis. So you're able to get your sample to results much more manageable amount of time, sometimes even within a day. So, so as part of the workflow for these molecular methods, one part, of course, is the sample preparation nucleic acid. And so we do have a MagMax kit specific for that, the MagMax Viral Pathogen Ultra Kit. Very similar to our MagMax Viral Pathogen kit, except it has an additional enzymatic digestion to help lyse the gram-positive bacteria as well as yeast and fungi. Like most of our MagMax kits, very flexible. It has a low input and a high input, either with the 96-well or 24-well format. Allows you to elute in a low volume, as well as use a multitude of different liquid samples. Is also compatible with all of our Kingfisher instruments, although we will focus the following slides on its use with the Flex instruments in this detection workflow. And so here you can see kind of how it fits in. So you have your different sample types, and for vaginal microbiota, of course, thin prep, e-swab are going to be your focus for things like the urinary tract, the urine, of course, even stabilized urine. So then these samples will be extracted with the MagMax Viral Pathogen Ultra Kit on the Kingfisher Flex. And so what it will allow is recovery of both RNA and DNA in your elution, including bacteria, yeast, virus, and protozoa. And then once you have this nucleic acid, then you can move on into our various TaqMan PCR panels. And these include up to the most high throughput, which is the open array, to more low throughput, which will be our singleplex TaqMan panels. Kind of to define those a little bit better, you can see, from low throughput all the way to the highest throughput. All of these are they have good sensitivity and specificity. It just really depends, if you want to maximize, the information you get, or if you only need certain information. So, but the benefit of this workflow, it provides you, and so here you can see we have several app notes focused around this. So looking at the vaginal microbiota, again, here's the workflow looking specifically with the open array. And so it's targeting these bacteria in a various number of formats, and it provides you just a large amount of data. And so here you can see, if you kind of break it out into specific research groups, you can see here's bacterial vaginosis, and looking at, to 96 samples, you can see it gives you the CT values, which then can be kind of worked with to find a pattern or to find, a specific detection target, for example, M. hominis. Same thing if you're looking at, here specifically aerobic vaginitis for these targets. And then again, looking more at Candida species for yeast. And then, of course, STI, if you're looking more for those. So it provides so much flexibility. And I think the final one is just the commensal lactobacilli within the sample. So a huge amount of information for relatively a small amount of effort. Similar we have for the urinary microbiota. Again, same workflow, just with urine samples, different targets. Again, we have an app note around this. But here you can see we actually looked at results with the open array with our TaqMan, which you'll see in blue, and then positive results generated by culture. And you can see again, the increase in sensitivity and detection for the PCR versus the culture. And then finally, here we're actually looking again, different targets, with culture versus the open array results. And you can see, again, how it compares against the culture. So equivalent or more sensitive results. And so with that, I will pass it on to Ketil, who will cover more of the protein applications you can perform with the instrument.
Thanks a lot, Emily. So that brings me to the final part of this training, that we will address or talk a little bit about protein application and protein application on the Kingfisher instrument. So I would like to talk a little bit about immunoprecipitation, and this is a method that are commonly used in many different research fields like immunology, basic cancer research, etc., which you would like to pull out the protein of interest or proteins that will bind to each other, so that you're studying protein-protein interaction, and these interactions are all quite often quite weak and transient interactions. And in such a workflow, then you typically use an antibody that will bind to your protein of interest, or the proteins that you would like to use to study protein-protein interactions. And then these antibodies then are conjugated to a solid surface, whether it's a non-magnetic or a magnetic bead surface. The magnetic beads being these Dynabeads that we have been working a lot with. And the downstream, to use it in downstream applications such as Western blotting or also mass spectrometry. The workflow itself includes many different steps. Here we have illustrated a direct approach, where you first functionalize your beads. In this case, it's the magnetic Dynabeads. So you functionalize the beads with antibodies, and this is due to the built-in advantages of the Dynabeads. The binding of these antibodies to the beads are super fast, so that can be done in like 10 minutes. And then you follow this with a washing step, where you remove unbound antibodies from the bead surface. So you're sitting there with antibodies that are conjugated non-covalently to the beads, before you then transfer these functionalized beads to a biological sample to pull out your protein of interest or protein complexes. And this is then followed by washing steps to remove unwanted complexes. And then finally, you elute off your target of interest or protein complex of interest for downstream analysis. At one point, we reached out to a number of researchers out there that are using this workflow and asked them what kind of pain points do they actually have with this workflow, and how could we assist them in making this this workflow even easier and better. And then the response from all these researchers were that this is a manual method, mistakes happens, but we would like to reduce the number of errors that we do in our basic research, just in order for us to make all the experiments that we do count, so that we do less mistakes. But we also would like to increase the reproducibility, so that we can compare our experiments better. But very interestingly, they also would like to have solutions that allow them to spend their time more wisely. So avoiding spending a huge amount of time in the lab doing pipetting, but more focusing on the science. That's, of course, a scientist's interest. So they would like to have hands-off solutions. And the solution is, of course, then the combination of the Dynabeads, the magnetic Dynabeads, functionalized for pulling out target proteins, and the Kingfisher instrument, which is, Suraj mentioned, a brilliant instrument moving beads instead of liquid. So this makes the perfect match to both reduce error rate, increase the reproducibility, but also to focus your workload in the lab by providing all the samples in your plates, and then just push the button and walk away, and then just come back when the protocol is done and the proteins that we have captured are ready for analysis. Of course, we could have done this very, very quick. We could have just transferred this manual protocol that we have developed over many years and just write a script in the binded software for the Kingfisher instrument. The software on this instrument is very intuitive, so you will be able to write this script within a few minutes. But we would like to take deep dives into this script that we have now have on the Kingfisher instrument, and just to make sure that the performance of this script is as good or even better than the manual protocol that we already have. So we developed an assay based upon acridinium orange that was conjugated to antibodies. And then we followed this labeled antibody throughout the whole protocol. So we conjugated antibodies that were labeled with acridinium orange on the surface of the Dynabeads, and then measured the signal on the beads, the signal that we were able to obtain in the washing buffer, and also in the elution or in the eluate, just to see the fate of this antibody. Because if we do lose them throughout the protocol, then we will also lose the target. And in order to optimize and fine-tune this script, we took a look at different parameters in this workflow just to see which of them were critical and which were not critical. And we looked at parameters like temperature, target volume, incubation time, washing steps and volumes, but also the elution volume and mixing conditions. And the result of all these experiments, and we did quite a lot of them, we ended up with 3 critical factors for a successful immunoprecipitation on the Kingfisher instrument. And the 2 of them were related to washing. So by having many washing steps and also with a more rough mixing conditions, you may run the risk of losing your target and the antibody from the bead surface. So then the yield will be reduced. So that's why we included very gentle mixing throughout the washing steps in this workflow on the Kingfisher. In terms of the elution, here we saw the opposite. So by increasing the elution volume, then you increase the yield. So we tested in the range of 30 microliters up to 100 microliters of the elution buffer. So the final slides, I would just like to highlight a few data sets where we compared the automated workflow, the ones that we have just optimized, testing all these different parameters, ending up with 3 critical ones. So here we have functionalized Dynabeads with antibodies and then exposed them to cell lysates. And then we have looked at all the proteins that have been bound to these beads, which we could then elute off by using low pH, like a gentle elution method, or more rough elution using LDS at 70 degrees Celsius. And then we have stained them using silver staining. So here we can see that whether we use low pH as an elution method or LDS as an elution method, we see comparable proteins bound and eluted off the Dynabeads, whether we use a manual or an automated workflow. So these 2 methods are equally good in terms of background or unspecific binding. But of course, the most important part is, of course, the yield. So we have then also performed the same experiment, but then we, instead of doing cell staining to stain all the proteins that are on the gel, we continue to do Western blotting to detect specific proteins that we have captured. In this case, we have captured the tetraspanin protein CD9 using either Dynabeads protein G or a Dynabead that have been conjugated with secondary antibodies and then functionalized with anti-CD9 antibodies. And also here, in terms of the bands, we can see that the yield of the CD9 is equally as good with automated versus manual. So in terms of the performance here on the immunoprecipitation, the automated protocol is at least as good as the manual protocol. But also the benefit here is that you can actually just push the button and walk away and do something more scientifically interesting than to do pipetting. So the final slide here, I would just like to conclude this session by showing the new Kingfisher Apex instrument, which is a really nice instrument that have, of course, all the benefits that are included in the Kingfisher Flex instrument, but also have added new features such as UV light. It's got barcoding, so you can scan the barcodes on the plates. It's also a cloud-based system. It's got a very intuitive touch screen that you can actually play around with the protocol if you need to do that on this screen. But as we are working in my group, we are working with cells. We are working with exosomes, with virus, SARS-CoV-2 virus. We are working also on proteins. I really like to. I'm very happy to see that this instrument has also got cooling, which is very beneficial when you do cell isolation on this instrument. And with this, I would like to thank you.
Thank you, Suraj, Emily, and Ketil, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many of your questions as we have time for. So let's get started. Our first question is, if I wanted to switch between applications, such as moving from DNA extraction to protein isolation, is this a difficult process?
Hello, can I answer this?
Sure.
Yeah, thank you. Okay, so it's actually really simple. So if you're doing any type of DNA extraction and you're using our kits, you would be familiar already with the software and how it all works in loading the plates. So if you move to a protein isolation, the only thing you need to do is select the right type of bead. So Ketil talked about some immunoprecipitation protocols and then some of the beads that we'd be using for that. So the really nice thing is if it's the Thermo Fisher bead set, there should be some protocols already written for the Kingfisher. So depending on the instrument that you have, the Flex or the Duo Prime, you can just download them from the website and then start using them straight away. If you have the Apex, which is cloud connected, you have access to them straight away. So it's actually really simple, and the process would be the same: loading the plates, adding them onto the machine, and moving it through. And it's the same for any of the applications. If they're Thermo Fisher kits, we should have protocols already, and if not, we've got a whole team of specialists that can support you in making sure that you get the right protocol and hopefully help you optimize the protocol as well.
Thank you. Our next question, You mentioned saliva. Do you have a recommended collection storage method for this sample type to best preserve all nucleic acids or cells present?
I can take this one.
Okay, great.
It really does depend kind of on your downstream application. There's a multitude available. And as I mentioned, there's both raw collection and stabilized collection. We do have a line of saliva collection devices recently released. This is our Specimax devices, and we offer both a raw version and a stabilized version, which is what I would recommend.
Great, thank you. Our next question is, can you put the Apex slide up again? It was only up for a second on my end. Let's see, I think if one of the presenters would like to go back to the slide with the Apex. Great, thank you. So then our next question is, what is the full workflow time, hands-on and instrument, for both the vaginal and urinary tract workflows?
I can take this one.
Great.
For the full workflow from once you have the sample till you're able to get results off the instrument is 8 hours or less.
Right, great. Thank you. And it looks like we have time for one more question. Who should I contact to determine if a process I'm currently running could be adapted to run on the Kingfisher system and to identify which would be best to try?
So I can answer that. You would need to contact one of our TSS teams because you're trying to adapt one of your current protocols, right? So that would need a bit more of a technical conversation. I don't know if we are able to get the details of this customer, and then I can make sure that someone gets in touch. Is that possible? Yeah.
Yes.
Okay. So if I could get the customer's details, and if possible, if you could let me know where you're based, because it depends on the right person we get to you. But just getting your contact details is enough, and we'll get someone to contact you directly about it.
Great. Well, thank you, Suraj, Emily, and Ketil. Do you have any final comments for our audience? All right.
Yeah.
Oh, sorry, Suraj, did you have something else?
Just to say thank you very much for your time. We really appreciate it. And any support you need moving your Kingfishers onto different applications, we're here to help.
Great. Well, thank you again, Suraj, Emily, and Ketil, for your time today and your important research. We would also like to thank LabRoots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.